So is my screen also visible to everybody? Is my screen visible? Can you see the screen? Just give a thumbs up or something if you are able to see my screen so that I know that uh, everything is fine. Yeah, okay, thank you. So now we'll start the session. So a very good evening. Okay, I think people are still joining. Okay, so a very good evening, everybody. Uh, Welcome to the first online doubt clearing session of RNA Biology course. So, before starting this session, a little introduction about myself. So, I am Apurva Soni. I am a PhD scholar from the University of Hyderabad, and I am a PMRF. So, as part of my PMRF TA-ship scheme, it's mandatory to take some classes. That's why I'm here, and that's why we'll be interacting throughout this whole class for about 4 or 5 months. So yeah, so what will be the way in which I'll be taking this session is that first we'll try to very quickly summarize the things that we have covered in the week, and I would expect you to read at least the lectures and just go through the lectures that have been updated on your profile. So today we'll be starting with our week 1, and subsequently in each doubt clearing session we'll be covering the topics of that week. So if you have already read, if you have already seen the videos, it's easier for you to understand. You will be able to grasp it easily, and you can also ask me questions if you have any. Okay, so I just wanted to know about you all. So you are all just a minute. I think your people want to, yeah. Yeah, fine. So you guys are like of which stream? You are bachelors or masters or like what? Just you can type in the message. So I just wanted to know who all the students are. Are you are like bachelor's students or master's students or what? Hmm? You can just type it in the box. So I want this session to be very interactive. It should not be like a one-way session where I'm only speaking and you guys are not saying anything. So because we'll be dealing some questions, so I would, I mean, just a minute. Yeah, so I would expect you to answer the questions. So for every question that I'll be putting on the screen, I'll be giving you 1 or 2 minutes uh, uh, depending on the question, and then you can after 1 or 2 minutes you can just write your answers in your message box. So but I think nobody's replying, so you guys are of graduation or your BSc, MSc, PhDs. So you guys all are, I'm assuming then you all are graduation students, BSc students, BSc or BTech students. Hmm? Because no one seems to reply, as if you have just joined the meeting and left. Is that the case? No? Okay. So I do not find this class as very interactive. We'll see. We'll see through the session. Anyway, so let us start with the course. So, week 1 was Introduction to RNA Biology and RNA World. Okay. So, in week 1, our major portion that has been covered is just the introduction, the basics of RNA biology, the basics of what is RNA world, who coined this world, how RNA evolved, how our genetic material throughout the time has evolved. So, what do you understand by RNA? Can anybody tell me what is your understanding about RNA? You can just type in the message box, or if you want to speak, you can unmute yourself and speak also. It's up to you. But please try to answer. Just say anything that you know. It need not to be perfect, okay? You just need to respond. So what do you understand by RNA? Anybody? Anybody from the class? What is RNA? What is the full form of RNA? Chalo. Starting from very basic. What is the full form of RNA? Nobody knows in the class? Nobody knows the full form of RNA? Hmm? Or nobody wants to interact, or everybody knows everything? So Kanan, Anbu, Kaleshwari, yeah, ribonucleic acid. Okay, so that means I have to say the names. Correct. Very good, Kanan. So he or she has exactly said it right, that uh, RNA, the full form of RNA is ribonucleic acid. So RNA has evolved through the period of time in such a way that earlier it started by assuming that it can be the genetic material, but then because of some problems that RNA has and some plus points that DNA has, ultimately DNA evolved as a genetic material. But we need to understand that RNA was the first self replicating biomolecule that existed even before DNA and proteins in early evolution. And to understand this, what we deal is about RNA world hypothesis. RNA world hypothesis. So, this phrase RNA world was coined by Walter Gilbert in the year of 1986. So, what this RNA world hypothesis tells us? This tells that RNA was the first self replicating biomolecule that existed, and it proposes that RNA based systems were capable of initiating the life and store the genetic information and also catalyze the biomolecular reactions. Okay, so first thing that you should know about RNA world is that RNA were the first self replicating biomolecules. Okay, RNA were the first self replicating biomolecules, and RNA has the capability of storing genetic information. Very important. It has the capability of storing genetic information, and also it has the ability of catalyzing the biomolecular reactions. So it can act as catalytic also. So this is essentially what RNA world hypothesis tells, that these are the self replicating RNA precursors which were even before in the evolution to DNA and proteins. Then RNA world means that RNA can perform both genetic and catalytic functions. Okay, so genetic function means that it can store the genetic information and it can transmit the genetic information just like DNA. So just like DNA can, okay, somebody wants to join. Okay, just a minute. Yeah. So just like, okay, fine. Well, oh God. So, just like DNA can store genetic information and transit to the next generation, similarly, RNA can also store the genetic information and it can be transmitted to the next generation. Then, catalytic function of RNA is very important. If you know about ribozymes are nothing but RNAs which acts as enzymes, and these RNA molecules can catalyze the biochemical reactions such as self replication, peptide bond formation, and therefore RNA can have both genetic and functional role. catalyst role, and also RNA is believed to have given rise to DNA and proteins. So DNA and proteins. So you all these 3 points essentially includes the RNA hypothesis, the self-replicating nature, the storage of genetic information, and the catalytic role. These 3 features essentially covers your RNA world hypothesis. So RNA has only uh, in due course of evolution given rise to DNA and proteins. So DNA are more stable than RNA, and proteins are more efficient in catalyzing the reactions than RNA, because we have enzymes. We all know that enzymes are what? Enzymes are nothing but proteins. So enzymes do what? They catalyze the reaction. So ribozyme, which is an RNA, can also catalyze the reaction. But normal protein enzymes are more efficient in catalyzing the reaction. Okay. So what I'm doing is that I'm not covering. See, we have just 2 hours and we have to solve questions also. So this is not actually my part, to be honest, to go through the theories. My part is only to solve some questions and that's it. But still, I'm covering some portion so that it becomes easier for you to relate, even if you haven't read, or even if you couldn't understand, you can ask me questions around. So we'll be just covering the main points of the different lectures that has been covered. Okay, so week 1 talked about essentially the RNA world hypothesis and some milestones of how the origin of life science has occurred. Okay, then in week 2 was about introduction to RNA biology and RNA world, the evidences. So what are the evidences that supports the RNA world, and that RNA world the building blocks? So to understand this, we have to understand the Miller-Urey experiment. This Miller-Urey experiment is very important. Okay, this is important not just for this course per se, but in general, if you are writing some competitive examinations. So actually, the my way of teaching is that I try to highlight the important topics, not which are important for your this course, but also for your some competitive examinations if you are planning to give CSIR, DBT, or whatever. So this Miller-Urey experiments can go into basic 1 or 2 marks questions. Oh, just a minute. Right. Okay. So, what is Miller-Urey experiment? Does anybody have an idea? What is Miller-Urey experiment? Any idea of what this experiment is about? What this experiment defined, or what this experiment proved? Anything? Any basic thing that you can think of? No? Hmm? Gayatri, Gayatri MBT. Do you know what is Miller-Urey experiment? Evangeline, do you know what is Miller-Urey experiment? Any idea about it? No? You must have studied about it, right? Nobody has studied it. Okay, fine. So this Miller-Urey experiment, which was conducted in 1953, basically provided evidence for how the organic molecules, which are actually the building blocks of life, could have been formed on early Earth through abiotic, that is, non-living processes. Okay, so this experiment was conducted by Stanley Miller and Harold Urey, and that is why this experiment is known as Miller-Urey experiment. Okay, so just a minute. Yeah, I'll just copy this image here so that I can explain you in a better way. Okay, fine. So basically, how was the experimental setup? Let us try to understand through this figure. So Miller and Urey, they tried to stimulate the conditions of early Earth's atmosphere, as in the conditions that were present in early Earth atmosphere. They have tried to replicate those conditions, having a closed system. So they had a flask which was filled with water, H2O, which was needed to stimulate the ocean kind of a condition. Okay. Then a mixture of gases which were thought to be present in early Earth's atmosphere, such as methane, ammonia, hydrogen, and of course, water vapors. You had electrodes to generate electric sparks, which mimics the lighting as an energy source. So, what are they doing? What they did? They tried to replicate the condition of early Earths. The lighting and the sparks were replicated by these electrodes. Then boiling water to stimulate ocean like conditions, then gases. Then this water in the system, it was heated to produce water vapors, and these water vapors stimulated evaporation. The gases were there which were stimulated, and electric spark was discharged to stimulate lighting, which provided energy for chemical reaction. And the gas mixture was then cooled, causing the components to condense and collected in a trap. So, what were the observations? The observations, this is the collected trap where everything was collected. Okay. So, what is happening? Water is boiling. Okay. This boiled water in vacuum, I mean, vapors are generated, other gases are also there present here, and then this goes here. There is a electrode connected which causes sparks, lighting effect. And of course, gases are there. This water vapor is going from here. And all these gases were allowed to condense. And then water containing the organic compounds were collected in this collection trap. Okay, so this lower chamber is representing your oceans, as I told you. So then, what were the key findings? The key findings were that several organic molecules have formed, including amino acids such as glycine and alanine, which are very much important for life. So, what they observed was that through these inorganic molecules that they have used, several organic compounds such as amino acids were formed. This supported the idea that complex organic molecules could arise from simple reactions under primitive earth like conditions. Okay, so simple reactions can ultimately lead to the formation of complex organic molecules, and this experiment provided a support for the theory of abiogenesis. What is abiogenesis? Can anybody explain what is abiogenesis? Abiogenesis means life arising from non-living matter. Okay. So this experiment showed for the first time that Earth's early atmosphere could have contributed to the formation of prebiotic molecules. That means the atmosphere that was present back in that time, that has contributed in the formation of prebiotic molecules, which led to the formation, or which led the foundation for the origin of life. Okay, and this inspired further research into prebiotic chemistry, which led to the modern studies of how our life has initiated. Okay, now there have been variants of this experiment which have continued to produce organic molecules. This experiment has been done in quite a many ways, and each and every way only gave them the idea that this experiment and this theory is certain and correct. So this is your Miller-Urey experiment. So I gave a little bit time to this Miller-Urey experiment because this is an important part, I feel. So then this is one of the experiments which were conducted, and in nature, what are the areas which are this is one of the evidences to show that RNA was the basic general origin material. Other, if you see that there are hydrothermal habitats, volcano and hydrothermal vents, and they generate extensive heat which provide a catalyst for biological reaction to happen. So these are all the evidences to see how our uh, life has been created on earth. Okay, this experiment was created to explain how life has been originated on Earth, and other evidences to support the origin of life in this way are the hydrothermal vents, the volcanoes, and the hydrothermal habitats. Okay, then other, if you go on a molecular biology level, then the ribosomal RNA ka phylogenetic tree, it has hyperthermophilic organisms, which again supports this hypothesis. So this I'm just giving a very brief. I'm assuming that you have already read all these things while you were doing the lectures. Okay, so now importantly we see what are the subunits of RNA. So RNA has three subunits. RNA, what are the subunits of RNA? It is a ribonucleic acid. It has a ribose sugar, a ribose sugar. It has a nitrogenous base, and it has a phosphate group. Okay, so these are the three subunits of RNA. Now let us talk in detail about the RNA world. So RNA world says that RNA can be stored as a genetic RNA can be used as a genetic material because it can store the genetic information. Then RNA can be a temporary copy of genetic material. Yes. RNA can be a carrier molecule. RNA can be a working part of a cellular machinery, and RNA can also act as a catalyst. And its example is ribozymes. Okay, this is the mirror experiment. So this is what was covered in lecture 2 of the first week's class. Then lecture 3 was about uh, the origin of monomers. I mean, if you have gone through the lectures, just try to understand the topics. Okay, it might not, you no need to get confused ki acha heading is this but it is not correlating with the heading, whatever. Just try to understand the topics and then you will be easy, it will be easier for you to correlate. Okay, so now RNA world, when we talk about the RNA world evidences, so what are the evidences that RNA exists? So the best evidence for RNA that it has existed since beginning is the presence of ribosomes and ribozymes. Okay, so ribozymes are the catalytic molecules and ribosomes are the organelles which helps in the protein synthesis process. But ribosomes and ribozymes both are made up of RNA. So RNA, ribozymes and ribosomes are nothing but a type of RNA. Okay, they are made up of RNA. Then other biomolecules or other molecules such as ATP, S-adenosine methionine, biotin, coenzyme A, they all are made up of RNA. And the prebiotic synthesis of sugars, that means the RNA, helps with the help of formose reaction, and borate helps in the selectivity. So just highlighting this because this might be important. That's why I just kept these two. Okay, this is important. Okay. Then polymerization of nucleotides helps with the help of phosphodiester linkage. That means what linkage is present in RNA? Phosphodiester bond. Phosphodi means two. So it's not a phosphoester bond, phosphotriester bond, phosphodiester bond. Okay, it's a two phosphoester linkages. And this is a condensation reaction, and it is the formation of a bond between the phosphate group of one nucleotide and the hydroxyl group of the sugar component. And then comes the Chargaff rule. So can these are the topics which were covered in the first day's class. Can anybody tell me what is Chargaff's rule? Anybody? Anybody who has any idea about what is Chargaff's rule? Hmm? Let me see. Krishnaveni, do you know what is Chargaff's rule?
No, ma'am.
Yes, you can say, or you can type in the message box also, whatever. is feasible for you? What is Chargaff's rule? Gen Z, do you know what is Chargaff's rule? Hmm?
Base pair of DNA, ma'am.
Krishnavini, I'm unable to hear your voice. Just a minute. I think. Yeah, can you speak again? Sorry, I think my system was in mute. Yeah, so can you say it again? Like whatever you said? Krishna? Hmm? Asiba? Anybody else? Kanan? Nobody knows about Chargaff's rule? So just tell me. Ki in a nucleotide is there. So A will pair with what? A will pair with G or C or T or U. A will pair with what? Hmm? A forms complementary base pairing with what? With which nucleotide? I want the answer. Just write it in the message box or tell me. What uh, with which nucleotide does A will pair? A will pair with? Why nobody is replying? This is very basic, no? You should know this. Why are you afraid of replying? Always, I've always had a class where students used to interact. See, I'm also a student, I'm not a teacher. So you can just, yes, good. So A will pair with T and G will pair with, it's easy. I don't think it even needs this much of time. G will pair with C. Yes, good. So Asiba and Anbu have replied very correctly. But A will pair with T in the case of DNA, right? In the case of RNA, A will pair with U. So this is what Chargaff's rule talks about, that A will pair with T and G will pair with C, and A number of A's should be equal to number of T's, number of G's should be equals to number of C's because they are pairing with each other. So this is what Chargaff's rule is about. That was yes, Kanan. Yeah, good. Now people have started replying. See, you just reply adenine with thymine, guanine with uracil. Okay, so adenine A will pair with thymine, adenine will pair with thymine, and G will pair with C, guanine will pair with C. A pairs with U in the case of RNA. In the case of RNA, thymine is replaced with uracil. Okay, so just making your concept clear that A will pair with T, but T is not present. I mean, T is not present in your RNA. RNA में uracil is present. So A pairs with U in the case of RNA. Okay, I hope it is clear. Now, in lecture 4 in week 1, now it's very interesting. So you see, um, DNA is stable than RNA. Now here we're talking about that we know that RNA is has a self catalytic excuse me. Yeah, so RNA has a self catalytic potential. Okay, and RNA acts as a genetic can act as a genetic material also. Then why there was a need of another genetic material? Why DNA has to come in picture at all? If RNA can perform both the functions, see, DNA is very I don't want to say the answer, but DNA cannot do the catalysis reaction. Okay, RNA has the ability to catalyze a reaction, but DNA does not have the ability to catalyze a reaction. But DNA can act as a genetic material, but that RNA can also do. Then why there was a need of DNA to come as a genetic material? RNA can do not. Not only the transmission of the genetic material, but it can also work as a catalysis. Then why over evolution DNA and not RNA evolved as the genetic material? Does anybody know why? Why DNA evolved as a genetic material over the period of time and not RNA? The answer lies in the question itself. Because DNA is more stable. That is why DNA in due course of evolution became the genetic material and not the RNA. Could you understand what I'm trying to say? Because RNA has this catalytic power, this catalytic power is there, but this catalytic power is making it more unstable, is making it hyperactive. Whereas DNA does not have the ability to catalyze a reaction, and that is why it is very stable. And we need a genetic material which is stable in a way that it can preserve all the genetic information from one generation to another. What if during the transmission of genetic information from one generation to another, some of the genetic material is lost because the carrier is only very unstable? It can happen, right? So, suppose if RNA is only the genetic material, and in the transmission of genetic information from one generation to another, since RNA is so unstable, it lost some of the portion, and that is very important. Important portion is only lost. Then who is at loss? Our replication, our transmission will become inefficient. So that is why if 100% of information is given to RNA, it should transmit 100% of information to the next generation. But RNA cannot do that because RNA is very unstable. It is highly reactive. So that is why DNA, which is not so reactive, which is highly stable, evolved as a genetic material because it can transmit 100% of the information from the parent to the offspring. Okay, so this is the reason why DNA and not RNA evolved as a genetic material. Now, on the molecular basis, there has to be a molecular basis, there has to be a science and logic behind how DNA became more stable than RNA. And what is that science? DNA has deoxyribose in the second carbon. The second carbon of DNA has deoxyribose, whereas, am I speaking correctly? Hmm? Just a minute. Nobody is listening, I think. Anyways, the second carbon of DNA has what? OH or H? The second carbon of DNA has OH or H. Do you know this? Does anybody has any idea about it? Hmm? The second carbon of DNA has only H, whereas the second carbon of RNA has OH in it. Okay. See, this is RNA and this is DNA. We are talking about the second carbon in their structure. RNA has an OH and DNA has only H. Because DNA does not have the oxygen, therefore it is known as deoxyribose. That means oxygen is absent. Whereas it has the oxygen, therefore it is only known as ribose. Ribose means sugar. So it's a sugar which does not have oxygen at the second carbon. It just has the hydrogen, whereas it is a normal sugar. And because DNA does not have oxygen in the second carbon, therefore it becomes less reactive because it does not have the oxygen has the electrons which can interact with each other, interact with the other molecules. So therefore it becomes less reactive and more stable because it does not have oxygen group in the second carbon. This makes DNA more stable. Then DNA cannot form any secondary structures also. What are secondary structures? Does anybody know what are secondary structures? Yes. So GP Muthu has rightly said OH. Okay. So anybody who knows what is the meaning of secondary structures? Nobody knows the meaning of secondary structures. What kind of secondary structures are present in RNA molecule? Can you tell me? What kind of secondary structures are present in RNA molecule? RNA has what kind of secondary structures? Nobody knows the answer? Okay. What do we call this structure? Hmm? This structure is known as, anybody knows? This is a hairpin structure or a stem loop structure. Okay. This is a hairpin structure or a stem loop structure, and this is nothing but a kind of secondary structure. So these kind of structures are not present in DNA, right? These kinds of structures, secondary structures, are only present in RNA molecules and not in DNA. Okay, then so this is just to tell you that why DNA has become the genetic material and not RNA, because DNA is more stable and DNA uh, uh, is very much inactive, so it cannot catalyze the reaction, and it is too simple and stable. And DNA has the H at the second carbon, whereas RNA has OH at the second carbon, and therefore DNA is called as deoxy. Deoxy means without the oxygen. Deoxyribose, and RNA has only ribose. Then certain points that you should know, that whenever we have two strands, the template strand is the one which is the original strand. These are just the basics, you know, basics that you need to know. I'm just telling you the basics. So the template strand is the one which is the original strand, and the complementary strand is the one which has been newly created. Okay, so you understand the difference between the template strand and the original strand. If you have a double stranded, the template strand is the one which is the original one, and the complementary is the strand which is newly created, which is complementary to the second to the template strand. So when I talk about complementary, what is the meaning of complementary? Hmm? What is the meaning of complementarity in terms of molecular biology? Tell me. Anybody? Same replica. Okay. So it's good that you have tried to answer. What can you please elaborate? What is the meaning of same replica when I talk about complementary? Do you mean that a complementary stand means if A is there, then A should only come? T is there, then T should only come? Okay, or pair strand. Okay, let us try to understand that. Good, you have tried to respond. Thank you, Kanan. So I always appreciate people who try to answer. It doesn't matter you are answering it correctly or not. What matters the most is that you're trying to answer, you're trying to interact, and most importantly, you are awake in the class and you're listening to what I'm saying. Okay, so now let us try to understand this concept about complementary, template, and non-template. This is the original strand, which I call as the template strand. Okay, now look carefully. It has A, G, T, T, C, G. This is the sequence of the template strand, let's say. Or if you want, I'll make it U because we're talking about U. Okay, now what would be the complementary strand? Hmm. What would be the complementary strand? Will it be the same as this? Will it be AGUUCG? Will it be this? No, it will not be this. So, what would be complementary strand? Complementary strand means whose sequence is complementary to the template strand. So, what would be the complementary sequence? It would be U, C, A, A, G, C. Could you understand how what I'm talking about? This is the complementary strand. And if you take this complementary strand as the template and make a new strand, it would be A, G, U, U, C, G. So this would be identical to the first one, but this is complementary. So you understand? Template strand is the original strand. Complementary strand is the strand which is newly being created. Okay, so newly created strand is always complementary to the original strand. So I'm just explaining it very, slowly so that you can understand because these are very basics. Okay, fine. So in the fourth lecture, so first week had 6 lectures. Okay, so in the fourth lecture, we the fifth lecture covered how genetic material moved from RNA to DNA. What is the meaning of secondary structures? What is the difference between a template strand and a complementary strand? And RNA replication helps with the help of RNA dependent RNA polymerase. Then we come to lecture 5. So, lecture 5 typically covers about ribozymes. So, ribozymes are basically your RNA which works as a which has a catalytic behavior, which has a catalytic function, so it can behave as enzymes. Okay, so ribozymes basically works as a nuclease. So can you tell me a process where ribozymes works as a nuclease? Hmm? Where in molecular biology, where you find ribozymes working as a nucleus? Can anybody think? I'll be really happy if you can answer this question. Where ribozymes work as a nucleus in uh, molecular biology reaction, basic reaction. I'll give you a hint. Either it is replication or transcription or translation. In these three only somewhere ribozymes work. Now can you tell me? Transformation? No. Replication, transcription, and translation. Somewhere here only ribozymes work. Translation, okay. Where in translation can you further elaborate, Gayatri? Where in translation? Okay, chalo. Post transcriptional modifications. Can you tell me the name of three major post transcriptional modifications that happen in a cell? There are post transcriptional modifications that happen. What are those post transcriptional modifications that happens in a cell? Now I have given you even more hint. It is somewhere in post transcription modification. Think. Can you think of? Acha, tell me the name of the three post transcriptional modification that happens in a cell. Hmm? Can you think of it? First is 5' capping, 3' polyadenylation, and removal of introns. These are the three post transcriptional modifications that happens. So in the removal of introns, there your ribozyme plays role. We'll eventually see it also. So ribozymes works as a nucleus in the removal of introns during post transcriptional modifications. Okay, then a shortened form of a ribosomal RNA of tetrahymena has shown to be an enzyme. This is just an example which catalyzes the cleavage and ligation of various nucleotide chains. Okay. So, the RNA world hypothesis says that the prebiotic soup was composed of a multitude of components such as protein, nucleic acids, lipids, and carbohydrates. Now, shift from RNA to DNA. RNA was much safer from degradation in the hairpin loop form but couldn't sustain this structure because of the nature of RNA. So, basically, RNA is very unstable. But if RNA is present in its hairpin loop structure in this form, then it is little bit safer. It is little bit less prone from degradation. But in RNA cannot be present in this form always. So that is why there has to be a shift in the genetic material from RNA to DNA. And all these molecules form bond in the presence of energy. It is okay. So now you see, I have kept some of the screenshots just to explain you because I thought these are the important points. So the pressure was on RNA to become resistant to degradation. Okay. RNA was much safer from degradation in the hairpin loop form but couldn't sustain this structure because of the nature of RNA. So, therefore, one replicating RNA molecule had a writing error that allowed for a different thymidine nucleotide to replace the common uracil, and this one chain allowed the RNA to become hairpin loop and looped in the form of a double strand. So, basically, hairpin loop is formed because of this mutation. But the thing to note is that even though RNA is much safer from degradation when it is present in this hairpin loop form, RNA cannot sustain this structure for a very long time. Therefore, RNA to DNA has to come for the genetic carrier, genetic information carrier. So DNA was far more stable, which allowed for a decrease in mutation. So DNA was far more stable. Why? Although this likely slowed down the rate at which differentiation occur, it also dramatically decreased the chances that the molecule will be degraded. Okay, so when DNA became the genetic material, now there was less chances of the degradation. Therefore, DNA actually became the genetic material. Over time, this molecule began to differentiate and compete much the same way the early RNA molecules did. Some DNA molecules utilized the tRNA molecules to create strands of amino acids. Other utilized lipids to form strong outer barriers that were only permeable to the things that needed. That means that DNA has also evolved itself in such a way that it can carry out the basic functions which are expected from a genetic material, which not only includes the carrying of genetic information but also the synthesis of certain amino acids or uh, making membrane more rigid and strong. Okay, so this is what which was covered in lecture 5. Then lecture 6, typically lecture 6 you will see, I mean you must have seen, it includes key discoveries in RNA biology and their impact. It talked about ribosomes and the different types of RNAs that are present in nature. You have mRNA, tRNA, rRNA, snRNA, snORNA, scarRNA, miRNA, siRNA, guide RNA, effervescence RNA, transfer messenger RNA, and PB interacting RNA. So these many different types of RNA are present in nature. See, so many different types of RNAs are present. So I have tried to so this is how we'll try to do in each and every class. That first we'll try to cover the basics of, we'll just try to brief of that week's lectures. Okay, very brief. We'll just revise whatever has been covered, and it would be really helpful if you could come with some doubts. So I would really request the students, we'll do questions now. Okay, don't go anywhere. So just a minute. So it would be really great if you could come and study little bit, read about it so that you can understand it even better, and also you can ask your queries and doubts in the class. So now we'll be doing questions, right? So I'll put the question slide. I'll give you 1 or 2 minutes, and in that 1 or 2 minute, you can think of the answer, and then you can write here in the message box. So please try to answer. It's going to help you only. I'm not going to gain anything from it. And don't hesitate that you don't know what others will think or what I'll think, and you don't even know the basics. We all are here to learn, right? I'm also a student. You all are also student. Okay? We just have different teachers at different time. So please try to interact. I want this class to be very interactive. I don't want it to be a boring normal class. Okay? So okay, these questions are easy. Try to answer. Even if you don't, anyways, we are going to understand the concept and see the correct answer. Fine. So the first question is, a ribosome is a DNA component, ribonucleic acid, ribozyme, or peptide nucleic acid complex. So it's 7:52 right now. I will switch the screen in after 2 minutes. 7:54 I will switch the screen. So try to answer it. It's very easy. It's easy only. Try to answer it. 1 minute left. Hmm. Okay, now I'm switching the screen. Let us see what you have answered. Okay, good. Good that people have answered. So Kanan has answered ribonucleic acid, Gayatri has answered ribozyme, Muthu has answered ribonucleic acid, Hiyam Hamil has answered DNA components, Krishnaveni has answered ribozyme, and Joseph has answered RNA. I'm sorry if I'm unable to pronounce your name correctly. Okay, I might pronounce some names incorrectly. I'm sorry for that. So, okay, good that people have tried to answer it. Let us see what is the correct answer. So, a ribosome is a ribozyme. The correct answer is ribozyme. So, I mean, ribosome is a ribozyme because it has the catalytic activity. But if you are trying to answer it in a way that a ribosome is or RNA is also, it also has a component of RNA, then that is also correct because ribosome is actually an RNA. Ribozyme is also an RNA. Ultimately, ribosome and ribozymes both are RNA components only. They are actually their ancestor. I mean, their basic is RNA only. But the thing is that why ribosomes can be called more as ribozymes, because it also has the catalytic functions. So those RNA which has the catalytic functions are known, are those RNAs are ribozymes. So in that context, ribosome is a ribozyme. But essentially both ribosome and ribozymes are RNA only. So good that people have answered. Now we'll come to the next question. Yeah. Nitrogenous base can be formed from CH4, CO2, HCN, or protein. Okay. This is 7:56. After 2 minutes, 7:58, I will switch the screen. Mm-mm. Okay, I'll switch the screen after 2 minutes only. Then I'll see. Okay, 1 minute left. 1 minute more. Hmm. Okay. We are switching the screens now. Let's see. Okay. Many people have answered. Krishnavini has answered HCN, Joseph HCN, Buttu HCN, Gayatri hydrogen cyanide, and helium also HCN. So, very good. I think everybody has answered it correctly. So, the correct answer is nitrogenous bases can be formed from hydrogen cyanide. Very good. Now, let us see the next question. Good. RNA has a backbone made by peptide bond, glycosidic bond, hydrogen bond, or sugar phosphate bond. Okay. And you have 2 minutes. Right now it is 7:58. I will switch the screen after 2 minutes. Okay. Try to answer it. Hmm. 1 minute more. Answer on the message box only. Hmm. 30 seconds more. Okay. Switching the screen now. Let's see. Fine. Hmm. So Gayatri has answered sugar phosphate. Akansha has also answered sugar phosphate. Krishnavini sugar phosphate. Kanan sugar phosphate. Muthu sugar phosphate. And Hamil also sugar phosphate. So good, good. Everybody has answered it correctly. So RNA has a backbone which is made up of sugar phosphate. Okay. Peptide bond. Where peptide bonds are found? Hmm? Peptide bonds are present in? Tell me. Where are peptide bonds found? Peptide bonds are formed in proteins. Good. So, peptide bonds are formed between two amino acids, which makes a protein. Then, glycosidic bonds. So, because this is important between amino acids to make a protein. Okay. Glycosidic bond. Where is glycosidic bond formed? Mm. Saccharides. Yeah. So basically, glycosidic bonds are present in carbohydrates. Okay. So basically, it connects one carbohydrate to another group, which can be another carbohydrate, or it cannot be a carbohydrate. So glycosidic bonds in carbohydrates. What happened? Oh god, I don't know what happened. Anyways, so glycosidic bonds in carbohydrate. Hydrogen bonds. Where are hydrogen bonds formed? Tell me. Where are hydrogen bonds formed? Hmm. Water. Yes. Hydrogen bond is present in water. So basically, hydrogen bond is a bond which is an electrostatic interaction. Okay. Hydrogen bond is an electrostatic interaction between the hydrogen to a more electronegative element, basically nitrogen, oxygen, or fluorine. So basically, hydrogen bond is mainly between nitrogen or oxygen or fluorine. Generally, we talk about oxygen, we talk about OH bond, which is in hydrogen. So, and sugar phosphate is present in your RNA. So good. This why I just discussed it, because I mean it's good know if you know these things. These are very basics, but I don't know what happened to this. Why it's not working? Okay. Yeah, it's working now. I'll just decrease the thickness. Okay, fine. Okay, so now we'll come to the next question. Yeah. RNA world hypothesis was supported by meteoroids, ribozymes, montromolornate clay, or Leuca. Hmm. Tell me the answer. Right now it is 8-4. We'll switch the screen after 2 minutes. Okay? Try to think and answer the question. 1 more minute left. It's 8-5 right now. Hmm. 30 seconds more. Okay. Okay, now I'm switching the screen. Let us see. Hmm. Okay. So Kanan has answered ribozymes. Muthu has answered ribozymes. Hamil has answered ribozymes, and Gayatri has answered meteoroids. Okay, fine. So the correct answer is RNA world hypothesis is supported by ribozymes. So just to quickly revise, there are three major important aspects when we talk about RNA world hypothesis. What are those aspects? First is that RNA are self-replicating molecules. Okay, important hai. Whenever we talk about RNA world hypothesis, first is that these are self-replicating molecules. This is the first thing which is important. Second is that they can be they can carry the genetic information from one generation to another. Okay, so they can store and transmit the genetic information. It can store and transmit the genetic information. And the third one is that it has the catalytic function. These are the three main important points whenever we talk about RNA world or RNA world hypothesis. Now, in this question, if you see ribozymes, why ribozymes is the correct answer? Because ribozymes are the RNA that functions as catalytic molecule. These are RNA that functions as catalytic molecule. Okay, so therefore this ribozymes comes in the category of this point of RNA world hypothesis, that RNA has a catalytic function. So whenever we talk about the catalytic function of RNA, we always talk about ribozymes. Is that clear to everybody? I hope this question is clear to everyone. Okay, now we'll come to the next question. Hmm. Next question is, Formose reaction plays role in a amino acid synthesis, nitrogen base synthesis, sugar synthesis, or methane synthesis. Okay, I'll give you 2 minutes. Right now it is 8:09. I'll switch the screening 8:11. Okay, try to answer this question. Think and answer. Just try to attempt at least. We'll see whether it is right or wrong. We'll see that later. 1 minute more. 1 minute more. 30 seconds more. Okay. I think time is already up. Let me switch the screen. See what you have answered. Okay. So only two have answered. Why? anything. Anyways, so Kanan has answered synthesis of sugar, and Hamil has answered nitrogen based synthesis. Okay, so the correct answer is sugar synthesis. Okay, so yeah, so let us try to understand this Formose reaction. So basically, Formose reaction, it is a prebiotic chemical reaction that converts, from the name itself, Formose, that means formaldehyde, into more complex sugars, including ribosomes and some other pentose sugars, which are essential for the formation of nucleotides. So this reaction is significant when we talk about the origin of life theories, because it is a potential pathway for the abiotic formation of carbohydrates. So what we are saying, that it converts, Formose reaction, it converts formaldehyde, which is into more complex sugars, such as ribose, some other pentose sugars, etc. Okay, and therefore, because the starting material is formaldehyde, so it is known as a Formose reaction. And why we are studying about Formose reaction, why Formose reaction becomes such important here, is because this is this pathway is a very potential pathway for the formation of carbohydrate abiotically. So this is important because this is a potential for a pathway for abiotic formation of carbohydrates. So why we are uh, focusing more and more on the abiotic term, because when the life originated, then whatever has come, there was nothing, there was no life. So when there is no life, that means there is no living thing which was present. That means the life had to originate from whatever abiotic sources that were present there at that time. Correct? Because there was no life, there was no nothing as living that can be called. So how then life originated? Then obviously the abiotic factors which were present at that time, they have only contributed to the formation of life. And when we talk about life, what we talk about? We talk about the complex or the simple and the complex organic molecules which ultimately combined and evolved to form life on Earth. So we needed basic organic molecules. We needed proteins, lipids, carbohydrates, nucleic acids. And these protein, lipid, carbohydrates, and nucleic acids ultimately evolved and formed. And through evolution, we know how the life has originated. And amongst all these molecules, the first one to come was RNA. And that is why RNA becomes so important, because RNA is one of the key factors which has helped in the origin of life and the evolution of life as well. Okay, because of its high catalytic, not high, but catalytic power and self replicating power, and storing of genetic information feature, it has been possible that life originated on Earth. Is it clear? So the first week class essentially talks about why RNA is so important for us to study, and why RNA is so important in due course of evolutionary history, and how RNA has actually helped in the origin and evolution of life. So RNA world hypothesis is actually very much important, especially if you are more interested in this, how the life originated and evolution and all those things. And subsequently, this in RNA biology, this is which is a very important course which I believe you will be studying about the different forms of RNA, how RNA is synthesized, that means the replication of RNA. Transcription, we will study how RNA transcribes, and then what are the post transcriptional modifications that takes place. These are all very important topics, I'll tell you, for your competitions, even for your course, like whatever courses you are doing in your bachelor's or masters or whatever. These questions are these topics are very important for you. You know, these are very basics which will be keep on coming in your course again and again. The complexity may increase, but the basic and the base will remain the same. So it's a good opportunity for you all, that you're getting, you know, a chance to understand about the RNA transcription and the machinery and everything, basic molecular biology in short. And I'll try my best. And if so, that's why I'll be even happier if you come up with queries and questions that you have. Okay, anyways, let's come to the next question. Something that we have been discussing for a while now. So, Chargaff's rule says G is equals to C, A is equals to U, A is equals to G, or C is equals to T. So, I'll give you 2 minutes. Right now, it's 8:17. I'll switch the screen at 8:19. Okay? Try to answer the question. And please, I want a little bit more participation on this one. It's an easy one only. Hmm. Options might be a little confusing, so you have to think and then you answer it, okay? Don't rush to answer it. Think and then answer. Okay. You have 1 more minute left. Oh, no, 30 seconds only. Hmm. We started at 8:17, right? Yeah, it's 8:19 already. Okay, then I'll switch the screen. Let us see what you have answered. Okay. Hmm. Ma'am, can I get these questions? Okay, first of all, let's see what people have answered. So Joseph has answered A is equals to G. Okay. Kanan has answered G is equals to C. Hamil has answered U is equals to A. And Krishna has answered A is equals to G. Then Joseph has a query. Ma'am, can I get these questions? So for everybody, so as per the rules, I'll be uploading these lectures and the PPT also. Okay, so you will get the link from where you can get the PPTs and the recorded session. So these recorded sessions are will be made available to you via a YouTube channel. So if you just, I'm just writing my name here for your confusion. This is my name. Okay, so if you go on YouTube and you search, you'll get my name. And okay, I've pinned it. So from this name, if you search PMRF, like, okay, just a minute, I'll tell you the exact name also. Hmm. So, okay, this is the name. This is the name of the YouTube channel, of my YouTube channel, where I'll be uploading all the recorded sections, sessions of this particular course. I'll make a separate folder, and then from there you can see, because there are other lectures also. I have taken this course previously, so previous lectures are also there. Don't get confused. I'll write 2025 so that you don't get confused in it. Other than that, you will also be getting the PPT that I'll be sharing with you. I think there would be a link in some of the Excel sheet, I don't know where, maybe in that course page where you have all the recorded lectures. I think there you will have the link for the PPTs also. So do not worry about that. You will get the questions, you will get the recorded lectures also. That is not a problem. Okay, I hope it solves your question. Okay, so now let us get back to the question. So many people have answered many different options. The question is, Chargaff rule states G is equals to C, A is equals to U, A is equals to G, or C is equals to T. And the correct answer is G is equals to C. Why G is equals to C? See, if we talk about Chargaff's rule, it basically describes the fundamental principle of DNA based composition and pairing. So DNA is composed, or if we of adenine, thymine, cytosine, and guanine. Okay. First of all, you should know A stands for adenine. Okay, spellings important. Adenine. G stands for guanine. Guanine. T stands for thymine and not thymidine. Very important. It's very basic, but still people get confused every time. It is thymine, not thymidine. And C stands for cyto C. RNA में you have U, which stands for uracil. Okay, so these are the four nucleotide ATGC. Now, Chargaff rules basically tells about the complementary base pairing. Complementary base pairing means that A will base pair with T and G will base pair with C. So actually this is written wrongly. If you see, there are only two bonds there. So if you see, this is wrongly written. But anyways, this means that A will only pair with T. Adenine will always pair with thymine, and guanine will also always pair with cytosine. And between adenine and thymine, there will always be two hydrogen bonds. Between guanine and cytosine, there will always be three hydrogen bonds. So, just first tell me, then which would be a stronger interaction? AT interaction or GC interaction? Which interaction would be stronger? Can you tell me? On the basis of number of hydrogen bonds, can you tell me which is a stronger interaction? AT interaction or GC interaction? Which is a stronger bond? AT or GC? Triple bond. Exactly. That means GC is a stronger bond. Good, Karan. Yes, Gayatri. Good. GC. So GC is a stronger bond because it has three hydrogen bonds. Okay. So since A will always pair with T and G will always pair with C, and one thing more, that Chargaff's rule stands for a completely base pair DNA. That means a double stranded DNA which is base, this is a double stranded DNA, completely it is base paired with each other. So if A is pairing with T and it is a double stranded DNA, that means each and every nucleotide has paired with each other. This means that A has to be equal to T and G has to be equal to C. Achha, this is equal to sign. Okay, this is equal to sign, not the bond sign. Fine. So this means A has to be equal to T and G has to be equal to C. Only then it can be base pair, na? So that is why A has to be equal to T and G has to be equal to C. A cannot be equal to uh, C or G. And because Chargaff's rule basically implies on DNA, therefore we are not correcting this. A is equals to U because U comes in RNA. Okay. So this is important. from which we get to know that amount of A is always equal to the amount of T, and amount of G is always equal to the amount of C. A is equals to T and G is equals to C, because then only a DNA can completely base pair with each other. If one A is less than T, then one bond would be empty. This is not, then this Chargaff rule cannot be applied. Chargaff rules can be applied only when a DNA is a double stranded, completely base pairing with each other. Okay, so this is important. Now we'll come to the next question. Is it clear to everybody? Is this question clear to everybody? Chargaff rules question. I hope this is clear to each other, to everybody. Now we'll come to the next question. These are simple questions now. Hmm. Question is, hydrothermal vents were important to give rise to life forms. True or false? Hydrothermal vents were important to give rise to life forms. True or false? Right now it is 8:27. I will switch the screen in after 2 minutes. 8:29, I will switch the screen, okay? Try to answer this one. 2 minutes you have. 1 minute more. Okay, now I'm switching the screen. So let's see. Okay, so many people have answered. Nice. Okay. So Joseph has answered true. Hamil has also answered true. True. I'm sorry if I'm pronouncing your name incorrectly. Kanan has also answered true. Asiba has also answered true. Gayatri true. Krishnaveni also true, and Anubu is also true. Good. So everybody has answered it correctly. Hydrothermal vents are important to give rise to life forms. The answer is it is true. So here where we have studied about it, we studied about it here. So when we were studying about the Miller-Urey experiments, and when we were talking about the further evidences to support origin of life and how the origin of biomolecules occurred, we talked about two things. First is three things actually: hydrothermal habitats, volcanoes, and hydrothermal vents. So all these three, they generated extensive heat, which provided a catalyst for the biological reaction to happen. And what was this catalyst? This catalyst was nothing but the enzymatic part of RNA. RNA only acted as a catalyst for the biological reaction to happen. So anything which is producing a lot of heat, which is generating extensive heat, that heat is helping in the biological reaction to happen. So whether we talk about the hydrothermal habitats, or we talk about the volcanoes, or we talk about hydrothermal vents, they all generated extensive heat, which provided the catalyst for the biological reaction to happen, because heat can accelerate the rate of reaction. Okay, because heat is nothing but energy, and your energy can accelerate the rate of reaction. Because when we talk about a catalyst, what is the function of a catalyst? Catalyst essentially works, yeah, So Michael Jiva has also answered it correctly. True. Yes, good. So when we talk about catalysts, what is the function of catalyst? The catalyst function is to accelerate the rate of reaction. Okay. So without changing the products, the final product. So if A is the substrate and B is the product, the rate of reaction of A to convert to B will be enhanced if we provide a catalyst. But A will only form B. Even in the presence of catalyst also it will form B only. But the rate at which A will produce B or A will convert to B will be enhanced with the help of catalyst. So this means catalyst is kind of driving the rate of reaction without changing the reaction. So heat provides energy. So energy can drive or accelerate the rate of reaction. That means hydrothermal habitats or volcanoes or hydrothermal vents, because they generated extensive amount of heat, this heat has helped in this biological reaction to happen in a faster rate, and this faster rate is also millions of years only, but on the evolutionary time scale we're talking about. Okay, fine. So let us move to the next question. Yeah. So next question is, proteins are able to act as better enzymes than RNA. True or false? Okay. So 8:33 I'll switch the screen after 2 minutes. 8:35 I'll switch the screen. Meanwhile, you just answer in the message box. One minute left. Hmm. Just 1 minute more. Okay, now I'm switching the screen. Hmm. Somebody wanted to join. Okay. Subha Lakshmi, we have already covered 90% of the topic for today's class. But that's fine. You can go through the recording, but I would uh, it would be better if you could, if you can join the class little early. Of course, I understand that you all have your classes and assignments and presentations and everything, but it's better for you if you join the class the right time. Anyways, so let us see what you all have answered. Joseph has answered true. Hamil has answered false. Kanan has answered false. Gayatri, where it went? Yeah, Gayatri has answered true. Krishnaveni has answered true. Anbu has answered true. Michael Jiva has answered true. Joseph has answered true and Hamil has answered false. Okay. Okay. Hamil has answered false. Gayatri has answered false. Okay. Good. Many people have attempted the question. Now, let us see and we'll try to understand also because there are many different answers. Proteins are able to act as better enzymes than RNA. The correct answer is true. Proteins are better. RNAs are also RNA also have catalytic behavior, but proteins are better in enzymes per se than RNA. And that is why whenever we talk about enzymes, whatever enzymes generally that are present in nature in your biomolecular reactions or your pathways or your like we study about the Krebs cycle, pentose pathways and xyz, wherever we talk about enzymes, all the enzymes that we talk are protein in nature. Protein. So can you tell me that why proteins are better as enzymes than RNA? Any idea who has answered it true? Can anybody tell me that why protein are better as enzymes than RNA? Hmm? See, first of all, you should understand that proteins are very versatile in nature. When I talk about versatile, how many different types of proteins are present? There are 20 different types of amino acids. Okay, there are 20 different types of amino acids. 20 different types of amino acids, they can be incorporated into many different ways. So there is much higher, greater diversity of chemical functionalities as compared to the same RNA. Could you understand that proteins, because proteins offer greater diversity as compared to RNA? This is first thing, because there are 20 different types of proteins, amino acids, not proteins. 20 different types of amino acids are present. So these 20 different types of amino acids with their own permutation combinations can generate high diversity. So chemical diversity is very high when it comes to proteins. Why? Because these 20 different types of amino acids, do they have the same chemical properties? No. Some are hydrophobic in nature, some are hydrophilic in nature, some are charged. So there is so much chemical diversity which is present in these 20 amino acids. And these chemical, because there is so much of chemical diversity which is present, therefore it can cater to a larger different variety of reactions. Some reactions might need hydrophobic nature of the proteins, some reaction might need the hydrophilic nature of proteins, some reactions might need the charged nature of proteins. So it can cater a larger family of reactions because of its chemical diversity. This is one of the reasons. Then the protein have a very complex 3D structures, and this complex 3D structure can allows them to specifically and precisely bind to the substrates. Therefore the catalysis is even more precise because of the complex 3D structure. Okay. And again, ultimately, that they have greater catalytic potential. The catalytic potential, only if just we compare the catalytic potential, proteins have a greater catalytic potential. Therefore, in due course of evolution, proteins have evolved as enzymes. Key most important point is because of its chemical diversity and the fact that there are different types of amino acids present, which can cater to a larger audience. Okay, so is it clear to you that why proteins are better than RNA when we talk about as enzymes? I hope this is clear to everybody. I hope who have answered it incorrectly now understands that why it is it so. Okay, fine. Let us move to the next question. I think we just have 2 3 questions more. Hmm. Prebiotic world is a soup of DNA, proteins, and lipids. Is it true or false? Right now it is 8:41. I'll switch the screen at 8 after 2 minutes. 8:42. Okay, I will switch the screen at 8:44. Think and then try to answer. Hmm. Try to answer it. 1 more minute left. 1 minute left. Hmm. Okay. Shall I switch the screen now? Okay, I'm switching the screen now. Let's see. Okay. Just a minute. Hmm. Okay, only 3 people have answered it. Hmm. Kanan has answered true, Hamil has answered true, and Muthu has answered false. Okay. So the question was prebiotic world is a soup of DNA, proteins, and lipids. And the answer is false. Now, why it is false? Why it should be false and why it should not be true? Think, what is this course? This course is about RNA biology. So, what we are saying from the start? We are saying from the start that the life originated, the one of the first molecules that was present when which led to the origin of life, or when the life originated, was RNA. And later on, in due course of evolution, DNA came into picture. Okay, so when we talk about the prebiotic world, prebiotic, before the life actually originated, it was a soup of RNA and not DNA. Okay, it was RNA and proteins and lipids that were present, not DNA. DNA came into picture much later. First, RNA was only the genetic material. Because RNA was so unstable and because of its catalytic behavior, it couldn't carry the complete genetic information from one generation to another. Then later on, with some modifications and with some evolution, came DNA. And then, because DNA is much more simple and stable, therefore DNA was chosen as the genetic material and not RNA. But the evolution, but the origin, when the origin of life happened, at that time it was all about RNA, not DNA. Even RNA is even before than proteins and lipids. First there was RNA and then proteins and lipids came into picture. Okay. So the correct answer is prebiotic world is a soup of RNA, not DNA. Now, easy RNA can store genetic information, true or false. 2 minutes. Right now it is 8:46. I will switch the screen in 8:48. Okay? Try to answer. This is very easy. One more minute. Okay. Okay, I'm switching the screen. Fine. Okay. So, Kanan has answered true. Hamil has also answered true. RNA can store the genetic information. So, yes, true. We all been studying that RNA can serve as a. Just a minute. What happened? Don't know. Hmm. Okay. Now, next question is. Oh, this was the last question. Fine. So, now we have completed all the questions. So, very quickly, let us see. So we started with the about RNA world, that what are the three properties that makes the RNA a world hypothesis: its self-replicating property, its ability to store genetic information, and the catalytic behavior. Then you go through the lectures and you will see some milestones which are important to be studied when we talk about the origin of life. Then we talked about the Miller-Urey experiment, how Miller and Urey showed that life originated with the help of this I've explained it already. So this building blocks of biomolecules, which are very much important, how they were created with the help of electric discharge and with the help of water and with some gases. And then some of the evidences, early evidences which supports this theory, were the hydrothermal habitats, volcanoes, and vents. And then the three subunits of RNA, we know that ribose, phosphate, and the nitrogenous bases. Now, RNA world essentially states that RNA can store genetic information, and it can be a carrier molecule, and it can also works as a catalyst. Then some other evidences that suppose that RNA was present much ahead and before is the presence of ribosomes, ATP, S-adenosine methionine, that is SAM, biotin coenzyme A, etc., and the Prebiotic synthesis of sugar took place with the help of Formose reaction, and prebiotic synthesis of purines took place with the help of hydrogen cyanides. And then phosphodinucleotides, phosphodiester linkage is present when we talk about the nucleotides polymerization. Then we talked about the Chargaff rule in detail. Okay, then we saw that how there was a shift from RNA to DNA when we talk about the genetic information and its carrying. That how, although it started with RNA, but how later on DNA took over the place and became the genetic material and not the RNA. And then in detail we talked about what is a template strand. Template strand is the original strand, and complementary strand is what? Complementary strand is the strand which is being created. So complementary strand is complementary to the template strand, not the original one. This is your template strand and this is your complementary strand. Then again we talked about the same things, ribozyme is very much important to study, and how there was a shift from RNA to DNA. Always remember, the stem loop structure of RNA is stable, but RNA is not present in this stem loop structure naturally, and therefore there is a shift from RNA to DNA. Okay, then some of the key discoveries that happened in RNA biology and the different types of RNAs which are present. So this essentially covers the just a minute. Okay. So this essentially covers most of the topics that we have that were present in week one's class, and then we did the questions. So this I just wanted to ask you: is this the way fine, or you want just the questions, or you want me to take a little bit revision of the theory of that week also or not? Because it's this class is for you, so I just wanted to know your opinion on it. Whether you want just the questions, or you want first the revision of the theory and then the questions. Which way? Either way is fine for me. It depends on you. Hmm? What you want? Nobody is replying. Anyways, if there is no preferences from you, then I'll take first the theory and then I'll take this thing, some questions, because I want you to understand the, just revise the concepts which are present and then go through the questions. It is easier for you to revise and understand the concept, and if you have any doubts you can ask me. Okay, so fine, then we'll stop the class here and we'll meet next week. And next week I hope that you will study week second lectures and try to solve your assignment questions, so that when we are solving some questions and revising here, it's even better for you to understand the concepts. Okay, fine then. Bye everybody, we'll stop the class now. Okay, you're welcome, Hamil and Kanan. Okay then, bye.